the question that haunted medicine for centuries. For hundreds of years, there has been a question that has constantly troubled the medical community. If cancer is an insatiable voracious eater, can it be killed by starvation? This question sounds simple, because if cancer grows thanks to the body's nutritional source, then just cutting off that source of life will cause it to weaken and die. It seems like obvious logic, but in reality, the journey to find the answer has lasted for more than a century, with countless experiments, failures, and even tragedies. And to understand that journey, we have to go back in time, to the early 20th century, when a German scientist conducted one of the most shocking experiments in the history of medicine. That person was Otto Warburg. Otto Warburg, the man who shed light on the voracious nature of cancer. Otto Heinrich Warburg was born in 1883 in Freiburg, Germany. He came from an intellectual family and from an early age showed a special passion for biology and chemistry. After World War I, Warburg returned to the laboratory and began a scientific journey that completely changed the way humanity understood cancer. At that time, most scientists had only a vague idea that cells needed glucose sugar and oxygen to survive. But how cells converted sugar and oxygen into energy remained a mystery. Warburg developed special oxygen measuring devices, called manometers, to track how much gas the cells absorbed. He began comparing normal cells with slices of tumor tissue taken from patients, and the results amazed him. Classic experiment: cancer doesn't craving oxygen. In the Petri dish, normal cells were breathing oxygen regularly. The measurements were exactly what Warburg had seen before. But with cancer cells, there was almost nothing. They barely used oxygen. At first, Warburg thought it was an experimental error. Maybe the culture solution was lacking nutrients. He added more glucose. The results were even more surprising. The tumor not only didn't use oxygen, but also produced a huge amount of acid. Warburg used the indicator phenol red, and the solution immediately turned bright yellow. He analyzed it and discovered that it was lactic acid, the product when our muscles work too hard and burn sugar without oxygen. In other words, cancer was doing something extremely primitive, eating sugar in a grab-and-go manner, creating very little energy, releasing a lot of acid, but multiplying faster than ever. The Warburg effect. Cancer disregards efficiency. In normal cells, sugar glucose is broken down into smaller molecules through glycolysis, which are then transported into the mitochondria, the cell's powerhouse, to participate in the oxidative cycle. This process is extremely efficient. Just one glucose molecule can generate more than 30 ATP molecules, the energy currency of life. But in cancer cells, Warburg saw the opposite. They stop right after glycolysis, do not bother entering the mitochondria, do not use oxygen, and only generate a mere 2 ATP molecules for each glucose. The rest is expelled as lactic acid. If this process is likened to a factory, then normal cells are like an optimized factory, saving raw materials and generating abundant energy. Cancer, on the other hand, is like a primitive forge, burning all its raw materials very quickly, producing little but opening countless new furnaces, increasing its capacity by eating more. Warburg put forward a bold hypothesis. It is the failure of cellular respiration, the weakening of mitochondria, that forces cells to switch to this primitive path. And that is the cause, not just the effect, of cancer. This hypothesis, later known as the Warburg effect, has become one of the most important concepts in understanding cancer metabolism. PET scan, when we see the hunger of cancer. To verify what Warburg observed, scientists in the following century developed a revolutionary tool, PET scan positron emission tomography. The principle is very simple. The patient is injected with a type of glucose attached to a weak radioactive substance. As it travels throughout the body, the glucose is absorbed by the cells. The PET scan shows which areas are consuming the most. On the image, the tumor appears bright, even brighter than the brain and the heart, the two organs that consume the most energy. They are like furnaces that constantly consume glucose. And when the patient is successfully treated, for example after a few rounds of chemotherapy, the bright area disappears, a sign that the tumor has stopped eating. With the PET scan, Warburg was visually proven cancer is truly a disease of insatiable hunger. The anti-cancer diet craze. If cancer is so greedy for sugar, wouldn't the simplest way be to starve it by cutting sugar out of the diet? That idea has inspired a series of diet movements throughout the 20th and 21st centuries. People have tried everything: complete fasting, ketogenic diet cutting starch, increasing fat, macrobiotic diet, abstaining from milk, abstaining from meat. At first glance, the logic is convincing. Indeed, the ketogenic diet can lower blood sugar. Fasting also lowers blood sugar. But when put into clinical trials, the results are always disappointing. There is no clear evidence that these diets shrink tumors. There is no significant improvement in survival in patients. Why? Because the human body is an extremely intelligent system. When you fast, the liver immediately produces glucose from reserves or from other substances, such as amino acids. Blood sugar levels are maintained stable, because if they drop too low, the brain will starve first. And the tumor is even smarter. It does not just wait for glucose from the blood, but can also create other energy sources, or call in new blood vessels to draw nutrients from surrounding tissues. The result? You are exhausted, but the cancer is still alive. Tumor and fat. A dangerous love story when the answer is not in sugar. In the previous part, we witnessed the truth. Cancer is an insatiable voracious eater. It consumes glucose like a raging fire, leading people to believe that cutting off sugar will deplete the tumor. But decades of testing have shown that starving the whole body does not kill cancer, but only weakens the patient first. That forced scientists to ask another question. If we cannot deprive ourselves of glucose, does cancer have another secret source of life? And the answer shocked the entire research community. Fat. Fat, a huge energy treasure. In the body, fat is not just the soft flesh under the skin that makes us lose confidence when looking in the mirror. Fat is actually a huge energy reserve that evolution has given us. If blood glucose is like cash in your wallet, always ready to use, then fat is like a long-term savings account. 1 gram of fat contains up to 9 kcal of energy, more than twice as much as 1 gram of sugar or protein. The normal body uses fat sparingly, only when hungry for a long time or when doing prolonged physical activity. But cancer is different. Shocking discovery: cancer sucks fat from surrounding tissue. In the 1990s and 2000s, when modern microscopes and fluorescent staining techniques were developed, scientists began to observe tumors in more detail than ever before. They discovered tumors not only eat themselves, but also order surrounding fat tissue to sacrifice their energy source. The fat cells adipocytes near the tumor begin to shrink, losing their original plump shape, becoming flat fat corpses. Meanwhile, the tumor swells, with lipid droplets accumulating in the cytoplasm. This mechanism is called lipid transfer, the process by which fat from normal tissue absorbed into the cancer cell. In other words, the tumor is like an organized robber, forcing its neighbors to feed it with their own fat. Obesity, fertile ground for cancer. If cancer can take advantage of fat, what happens in obese people who have more fat than normal? Epidemiological studies have given a clear answer. Obesity increases the risk of at least 13 different types of cancer. Among them are breast, colorectal, liver, pancreatic, kidney, and even esophageal cancers. The reason is not only because there is more fat for the tumor to absorb, but also because fat is an endocrine factory. It secretes all kinds of hormones and cytokines, many of which can stimulate inflammation, increase blood vessels, and promote the growth of malignant cells. A typical example is estrogen, a hormone normally produced by the ovaries. But in obese people, fat tissue also produces estrogen, increasing the risk of postmenopausal breast cancer. Another example is leptin, a hormone that signals a feeling of fullness. Instead of protecting the body, Chronically high levels of leptin can stimulate cell proliferation and the formation of new blood vessels to feed tumors. The result? Tumors not only eat fat, but are also encouraged by fat to grow stronger. Tumors and collaboration with fat cells. Cell culture experiments have shown a strange scene. When cultured alone, cancer cells grow at an average rate. But when cultured together with fat cells, the reproduction rate doubles, even triples. Fat cells release free fatty acids EPA and lipid droplets. Cancer immediately absorbs them, transfers them to mitochondria, or stores them for later use. Furthermore, fat cells also secrete adipokines, signaling molecules that stimulate cancer to become more aggressive and metastasize more easily. As a result, in fat-rich tissues such as the breast, ovary, and pancreas, cancer often develops rapidly, is difficult to control, and spreads easily. Cancer and the fat truck through the body. Not stopping at sucking fat from neighbors, many recent studies show that tumors also know how to activate the whole body to transport fat to nourish it. The intermediary mechanism here is lipoprotein, the trucks that transport fat in the blood, such as LDL and VLDL. Cancer can increase the expression of LDL receptors, like building more docks for fat trucks to dock. This explains why in many cancer patients, blood lipids are severely disturbed. Instead of being used for the body, fat is taken over by the tumor. Cancer cachexia, when the patient shrinks while the tumor swells. One of the most frightening manifestations in late-stage cancer patients is cachexia, a condition of severe weight loss, muscle loss, and fat atrophy, while the tumor continues to grow. Part of the reason is that the tumor secretes factors that cause fat and muscle tissue to break down, releasing fatty acids and amino acids to feed itself. In other words, cancer eats the body from the inside. No matter how much the patient eats, he or she cannot gain weight, because all nutrients are raped to feed the tumor. Experimenting with fat metabolism, when they understood that cancer depends not only on sugar, but also on fat, scientists began to try new approaches. Fatty acid synthase (FASN) inhibition. Many cancers not only use fat, but also synthesize their own fatty acids. FASN inhibitors have been tested in animal models. Tumors shrink. Blocking fatty acid transport CPT1 inhibitors. CPT1, the gateway that brings fatty acids into mitochondria to be burned for energy. When blocked, cancer cells lack energy and their growth rate slows. Statins, cholesterol-lowering drugs. Some studies have observed that patients taking statins to treat high cholesterol have a lower incidence of cancer, or the disease progresses more slowly. However, the results are still controversial. White fat, brown fat, and open questions. Not all body fat is the same. There is white fat, which mainly stores energy, and brown fat, which is rich in mitochondria and specializes in generating heat. Interestingly, cancer seems to prefer white fat because it is a huge energy store. But new studies are asking, is it possible to convert white fat into brown fat, making it less susceptible to being exploited by tumors? If it can be done, it could open up a whole new direction of treatment. Blood, the red river that supposes death blood, the center stage. If the body is a country, then blood is the river that carries oxygen, nutrients, hormones, immune signals, everywhere. No cell can survive if cut off from the blood, and cancer, a rebellious part of the body, is no exception. But cancer does not just passively receive blood. It hijacks, transforms, and turns blood into its own tool. Tumors, architects of vascular cities. In the 1970s, Professor Judah Folkman, USA, came up with a shocking idea. Tumors cannot grow more than a few millimeters without their own blood vessels. When first formed, a cluster of cancer cells is just like a speck of dust, surviving on nutrients that permeate the tissue. But to become a real tumor, it must build a traffic infrastructure, that is, stimulate the growth of new blood vessels. That process is called angiogenesis. Cancer cells secrete VEGF, vascular endothelial growth factor, a powerful signaling molecule that causes nearby blood vessels to sprout new buds, stretch out, and wind their way into the tumor. The result? The tumor is like a city with an exploding population, immediately building a dense network of roads to transport oxygen, sugar, and fat. But those are not normal blood vessels. The blood vessels in tumors are far different from healthy blood vessels. They are crooked, leaky, and disorganized. The vessel walls are thin, fragile, and the pressure is unstable. This creates two consequences. The tumor receives continuous nutrition, despite the chaotic flow. Treatment drugs have difficulty penetrating deeply because leaky vessels increase tissue pressure, hindering the drug. In other words, cancer not only builds roads, but also turns roads into mazes to protect itself from attack. Blood, the road for metastasis. What is more frightening? Blood not only nourishes cancer, but is also a highway for metastasis. Cancer cells can infiltrate into the blood vessels intravasation, drift along the bloodstream, and land in a distant organ extravasation. This journey is arduous. To 99% of cells floating in the blood will die due to the pressure of the flow or be destroyed by the immune system. But if only 1% survive, it can create a metastatic nest, and at that point, the cancer enters a stage that is almost impossible to cure. This is the reason why most patients die not from the primary tumor, but from metastasis. Blood, the immune battlefield. Blood not only carries nutrients, but is also the patrol route of the immune system. T cells, NK cells, macrophages, constantly scan and hunt for foreign cells. Initially, many cancer cells are destroyed as soon as they arise, but over time, the survivors learn to hide, reduce MHC expression to avoid recognition, secrete TGF-beta, IL-10 to weaken immunity, attract TREG T regulatory cells, the internal police cells that silence the immune army. Result: the tumor turns the bloodstream into a biological shield, where immunity is lulled to sleep. Cancer and blood, a strange symbiotic relationship. Cancer not only sucks nutrients from the blood, but also reprograms the entire blood metabolism, increases blood glucose used for glycolysis, increases free fatty acids in the plasma, disorders lipoproteins so that more fat is transported towards the tumor. Even platelet alteration. Cancer cells hide in platelets to escape immunity when traveling. This is an evil alliance. Blood is both a carrier, a shield, and a stepping stone for cancer to travel far. Anti-angiogenesis trial. When Folkman announced the idea of blocking blood to kill cancer, he was initially ridiculed. But then, in the 2000s, the drug bevacizumab was born, an antibody that inhibits VEGF. It created a turning point. Many patients with colorectal, lung, kidney cancer, lived longer thanks to the drug that blocks angiogenesis. But the joy did not last long. Cancer quickly found a way to overcome, creating angiogenesis by a mechanism other than VEGF, using existing blood vessels instead of creating new ones, causing the phenomenon of pseudoangiogenesis. Cancer cells line up themselves into fake tubes. The result? Anti-angiogenic drugs only prolong the time, but rarely eradicate it. Blood, the mirror of cancer. In the past decade, another revolution has taken place. Instead of just looking at tumors through biopsies, doctors have begun analyzing blood, where cancer leaves its traces. ctDNA, circulating tumor DNA, fragments of cancer DNA that fall from tumors and float in the blood. CTC, circulating tumor cells, intact cancer cells on the move. Exosomes, small bags containing RNA, proteins that cancer secretes into the blood. These traces help detect cancer early, monitor treatment response, and predict the risk of recurrence. This is called a liquid biopsy, one of the biggest advances in modern oncology. The future. Can blood be turned into a weapon against cancer? Researchers are trying a number of bold ideas. Boosting immunity in the blood, using checkpoint inhibitors PD-1, CTLA-4 to unleash T cells, reprogramming platelets, making them attack rather than protect cancer. Nanorobots in the blood, nanoparticles designed to find and destroy circulating cancer cells. Hemodialysis against cancer, like dialysis, but the goal is to eliminate ctDNA or malignant cells. It sounds like science fiction, but many studies are coming out of the lab. 3 sources of life, 1 big picture. We have come a long way. Part 1, cancer, the sugar glutton. Part 2, cancer, the dangerous lover of fat. Part 3, cancer, the usurper of the blood river. From sugar to fat, from blood vessels to immunity, cancer emerges not as a mass of out-of-control cells, but as a malignant ecosystem, exploiting every resource to survive. And the paradox is, because cancer is so deeply dependent on metabolism, it also reveals its Achilles heel. If we understand enough, we can turn the source of life into a fatal point. This war is not over, but each discovery about sugar, fat, and blood brings humanity one step closer to the day when it can cut off the source of death.